First and foremost, I want to welcome you all. I see a lot of familiar faces and it's so nice. And I just really want to thank each of you for continuing to support MGFA and our efforts. And every time we have a webinar or host a call, I do see a lot of the same faces. So I really do appreciate that very, very much. Today is especially exciting. And for those of you who don't know me, let me introduce myself. I'm Sam Masterson, CEO of MGFA. And today is exciting for us because this session marks a new initiative for MGFA. We're going to start hosting a research series. It's called What's New in MG Research? And the reason we can actually do this is because there's more movement in the space than there ever has been. So that is certainly exciting, and it's only good for all of us in the MG community. So we do very much see it as our role to bring you the most current, timely research news on clinical trials and their respective outcomes. But we certainly will also be hosting webinars on other research-related activities outside of clinical trials when that is appropriate and when we have that information to share. So we certainly do not feel that our community members should have to go and search for good information. So we do see that as our role. Today, I am very proud to kick off this effort with Dr. Jeffrey Guptil. Dr. Guptil, is with Duke University School of Medicine. And Jeff also serves as the chair of MGFA's Medical and Scientific Advisory Board. This afternoon, Dr. Guptil is going to discuss Momenta Pharmaceuticals' Vivacity MG Study, as well as highlight information about the ongoing CARE-MG COVID-19 Registry. So welcome, Dr. Guptil, and thank you for helping us to kick off this research series.
Thank you, Sam. So welcome, everyone. Thanks for attending the session this afternoon. So as Sam mentioned, one of our goals here is to start, presenting and providing more information on trials that are happening to keep the patient community and their support system informed of what's going on in the field. And so we will continue to offer these sessions, and it looks like there probably going to be quite a few of these because there's a lot of movement happening right now in clinical trials, as Sam mentioned. So this will be the first in a series. And so to set the stage for this series of talks, one of the objectives for today is just to go over clinical trials a little bit so that people who haven't participated before can have a better understanding as to why we need clinical trials, how they're conducted, and just some of the key aspects of clinical trials in general. Then we'll kind of put that framework to the test and talk about a clinical trial that was recently completed by a company called Momenta Pharmaceuticals. And then at the end, I'm just going to mention one other effort that's underway for patients and how we understand COVID-19 and its impact on patients with myasthenia. And some of you may have heard me mention this a couple weeks ago as well. Okay, so to kick off with, we'll first talk about clinical trials. So, I think a lot of people have an impression of clinical trials in terms of, that the patients are involved in, and that we need patients to participate in them to get a drug approved to be able to use in the clinic. And while that's true, and a lot of the activity for clinical trials is critically important for getting a drug approved to give to patients, I just also want to emphasize that there's a lot of work that happens even before you get to the clinical trial stage. And this work can take years, or typically does take many years to come up with a target. What do we want the drug to do? What do we want it to hit? Then to test that drug in in several different types of models to make sure that it's not going to affect, a person's heart or their liver, to understand how it may interact with other drugs that a patient may take, and then to test it in animals, so that by the time it gets to humans, there's been years of data that's accumulated on a particular drug. And then this process of clinical trials, as you are probably aware, can take many years to complete before an application is submitted to the FDA or other regulatory agencies to get a drug approved and to make it onto the market. And so ultimately, the goal of this whole process is to find, the right drug, the right therapy that should be available to people. We want a drug that's clean, that doesn't have lots of off-target effects that could, cause problems. We want it to be as selective as possible. We also need to know, which patients should this drug be given to. So we need to figure that out as part of the drug development process, and in particular in clinical trials. We need to determine the dose. We need to figure out a dose that has the intended effect, that makes people better, but isn't too high that it can cause problems from it, or too low that it doesn't have the intended effect. And we also need to know the time to give it. So how often do we need to dose it? And is there a particular state or time in a disease course where it's the best time to give the therapy to a patient? So it's a very complicated and typically drawn out process to bring a drug to the market. Typically takes over a decade. And so a lot of you are probably following what's happening with COVID-19 vaccine development, at least to some extent. And you can see from this timeline here of over 10 years, we're trying to compress that into the span of a year or two, which is essentially unheard of. So yeah, there's a lot of, obviously work going outside of what's happening in MG that's, really important for all of us as well. So with that kind of background, what is a clinical trial? So, and this is more of a technical definition, but to simplify it down, we're testing an intervention on humans. So an intervention, which is typically a drug or other type of therapy, or it could be a vaccine. And we're looking to test the effects of that intervention on people. Does it make them feel better, or does it cause any problems that we can detect? And so, one of the main aspects of these clinical trials, particularly the later ones that patients become involved in, is that there's typically an active treatment drug, the drug that we want to test, and then another comparison group that either is not getting the active drug or is getting some other therapy that's being compared to. And so to really understand the data that's generated from a clinical trial, it needs to be really well designed and conducted. And for a number of reasons. One, so that we can understand the data at the end to make sure that it is, quote, the truth for what is being studied, but also to make sure that patient safety is being considered in the design and the conduct of the study, so that ultimately we can improve patients' health. So in general, when we get into patients or into people with trials, it's typically broken down into 3 phases, and you may have heard these terms before. So phase 1 are looking at often healthy volunteers, so people without a medical condition first, just to see if there are any major issues with the drug, and also to determine the dosing that would appear to be the most effective or the best to test in patients. And then in phase 2, these are typically the first studies that test a drug in a patient population. So for instance, patients with myasthenia gravis typically would get studied first here in phase 2. These studies tend to be smaller. And the goal of these is, again, oftentimes to look at a safety of the particular therapy in patients, and then to get a sense of what it looks like in terms of whether it has the intended effect in terms of efficacy. Does it help people? And that information that is generated in phase 2 is then often confirmed or expanded upon in phase 3. And so phase 3 clinical trials are oftentimes referred to as pivotal clinical trials. And these are the studies that the regulatory agencies like the FDA base a lot of their decision making on. So it tends to be a much larger population of patients that are being studied, and using the learnings from the phase 2 trials are incorporated into the phase 3 study design. So here you'll see that it's listed that there are hundreds or thousands of people that can participate in a phase 3 study. For a rare disease like myasthenia, it tends to be much smaller than that, on the order of 75 to a couple hundred. So here's in a generic form, here's what a design of a phase 2 clinical trial might look like. This is probably the most common design that's used in drug development. So we're intending to study the drug in patients with myasthenia gravis, and typically there's a visit or a period where we get background information on the patient and laboratory work, and also test to see how weak people are, to see whether they qualify for the study. And so there are typically a number of eligibility criteria that define whether or not a patient can participate in the study. So I've just given a few examples here. So they got to be over 18, they have to be an adult. They have to have a diagnosis of myasthenia. They have to have a certain severity of disease. We need to be able to measure something to see if the medication is actually helpful. And there are oftentimes medications or other medical conditions that we need to check for, particularly if there's a potential risk in a patient population that we don't want to expose to that risk early on. And then once we know that a patient is eligible, they're then randomized. And that means that they're assigned to one of the treatment groups. And I mentioned earlier that one of the groups tends to be the active drug that you want to study. And then the other one is either a placebo or another type of control. So a placebo meaning, they get like a sugar pill or a water injection. And then the patients are treated, and no one knows which group that they're in over the period of the study. And here I've just arbitrarily chosen 90 days. So for 3 months, they're in the trial, and they get the drug or the placebo. And then the drug is stopped, and then you have a couple follow-up visits after that to make sure that people are still doing okay and to get some additional assessments from them after that. Okay, so that's a little background on clinical trials. Now, I assume most everyone on this call is familiar with myasthenia and what causes it, but I think for this particular discussion, it's quite important to make sure we're on the same page. So I'm just going to run through this really quick. As you know, myasthenia causes weakness, and that weakness is from a disruption of the communication between the nerve and the muscle. So if we want to lift our arm, a signal comes down from our brain, down the spinal cord, and then it goes to a motor nerve, which then makes contact with the muscle. And then in the muscle, or at the end of the motor nerve, it makes a lot of branches out to muscle fibers. And these little cylinders are the muscle fibers under a microscope. And these little branches are where the nerve communicates with the muscle. And this is where the problem occurs in myasthenia. And we're going to zoom in on this here in a little more detail. And so you know that myasthenia is most commonly caused by antibodies to the acetylcholine receptor and rarely to another receptor called MuSK, muscle-specific kinase. And these antibodies disrupt the communication between the nerve and the muscle. And so here you can see this is a normal motor nerve, and then this is what it looks like on the muscle. And we have the receptors, this acetylcholine receptor here, that's supposed to be here to receive the message that's released from the nerve. And in myasthenia, because of the attack on the muscle, we lose this nice architecture here that's considered normal, and the communication between the nerve and the muscle breaks down. And specifically, there's a population of immune cells called B cells that produce these antibodies, and these antibodies attack the receptor that's on the muscle. And these antibodies are a class of proteins called immunoglobulins, specifically IgG. And this will be really important as we move forward here. So this antibody attack on the acetylcholine receptor is something that we'll be giving information on in this particular presentation, but a lot of companies are investing a lot of time and money to impact this, the attack of the antibodies on the receptors. So now we're gonna talk about this study, which is called Vivacity. And the study is sponsored by a company called Momenta Pharmaceuticals. And for disclosure, I was a key investigator on this phase 2 study and served as a site investigator at Duke for this. So this particular drug, it works very high level. What it does is it tries to lower the IgG levels, including the acetylcholine receptor antibodies that are so key to the disease. And the life cycle of IgG and antibodies is really tied in to this, the neonatal FC receptor. And it really helps to establish the lifespan of IgG molecules, how long they persist in your blood. The longer that they're around, of course, particularly for the bad ones that we don't want, the acetylcholine receptor antibodies, the longer that they're around, the more damage that they can cause. And so Momenta's drug called nipocalimab, which I'm going to call Nipo from now on just to make it easier, is a drug that interferes with this life cycle of IgG. And the end result is that antibodies are destroyed or removed from the circulation in a rapid fashion. And there are other companies that are working on this type of mechanism that you'll probably hear about in the future, in future presentations. So this is a complex slide, but I'll try to run through it here briefly. So on the left is what's happening in the normal state. And then here, it will be the example for myasthenia on the right. So under normal circumstances, if this is the blood takes up IgG molecules, which are almost always represented in this Y form, because this is what they look like if you look at it under a microscope. And it takes it up into these little spheres, and they interact with that FcRn receptor that was mentioned on the previous slide. And if they bind to this receptor, they get spared and they end up going back into the blood. And so there's this recycling process that happens. However, if the IgG molecules don't bind to the Fc receptor, they get targeted to be degraded and removed. And so what Nipo does is if this is infused into a person, it competes for the Fc receptor here. And it's been designed to do a much better job at binding to the Fc receptor. So as a result, what happens is more of the IgG that's circulating in a person ends up being degraded and removed from the circulation. And very little of it ends up going back into the blood. And so you can think about how that could potentially be a helpful strategy for treating myasthenia. So if we can remove the antibodies, the bad antibodies from the blood, that means that they can't attack the muscle, and that hopefully patients' weakness would improve. We do have some precedent for this in myasthenia, this particular approach to treating myasthenia by removing the antibodies from the circulation. And so many of you may be familiar with plasma exchange or plasmapheresis. And that is basically the same concept, is that instead of doing it with a drug, we're removing the blood, filtering it to remove the antibodies, and then giving the blood back. And both have the same basic effect, to remove the antibodies that are causing the problem. So for this clinical trial, the study questions that were being asked are, first, is Nipo safe for myasthenia gravis patients? Can Nipo remove or reduce the IgG from the circulation, from the blood? And in particular, can it reduce the acetylcholine receptor antibodies? And finally, can we get a hint that it seems to help patients in terms of their symptoms? And so here's the study design, and you'll probably notice that it looks pretty similar to what I showed earlier. So there's a screening process where we check to make sure that patients were okay to be in the study, that they had myasthenia, they had enough myasthenia that we could measure it so we could detect a change. And we can get into some of the specifics on that in the upcoming slides. And then the patients were randomized. So again, flip of a coin to see which group they got assigned to. And for this study, rather than 2 groups, which I showed before, there were 5. So one group got placebo, and then the other 4 groups got the treatment, which at the time was called M281, and now it's called nipocalimab. So, but this is the same, it's Nipo. And so the different doses, there was a 5 milligram that you got every 4 weeks, a 30 milligram every 4 weeks, 60 every 2 weeks, and then 60 once. And so the idea from having these different dosing, these different arms, different treatment arms, was to get a sense of how much drug people needed to have an intended effect. And then there's also the possibility that you could give someone too much of the drug, and that would be important to know too. So was there a dose that caused any problems? And those doses were selected based on the data that they had from phase 1, where they're testing it on healthy volunteers, as well as some of their preclinical data before they got to humans. So they're treated for 8 weeks, and then they're followed for 8 weeks to see how long it lasts after they're treated, and then to make sure that there weren't any late problems that people were having. So the total study, the goal was to enroll 60 patients into the study. And then once people completed, there's an option to go into an open label extension phase, or open label extension study, where in this case, everyone would get the Nipo drug. So you can imagine, if you're participating in the study and you were just getting the placebo, it's very nice to be able to, at the end of it, for participating, you will also get the opportunity to get the active drug at the end. And so who could participate in the study? So this is just some of the key concepts here. So you had to be an adult, you had to have myasthenia, not surprisingly. And you had to have a severity of myasthenia that was either mild to severe at the start of the study. So, and this is a classification scheme that's commonly used in myasthenia trials. You had to have antibodies detectable for acetylcholine receptor or MuSK. And you had to be on stable medications to be able to participate. And that's really important because we don't want to, if patients' medications are changing during the course of the study, that could impact their clinical status, how well they're doing. if they change, it would become very difficult to interpret what's the effect of the study drug versus what's the effect of these other changes in their myasthenia meds that have been made. So that's why it's really important that those medications need to stay stable. And they had to have a minimum symptom burden and amount of weakness that are measured on these scales to allow them to be eligible for the study. And that's to help make sure that there was that if we could measure detectable change over time with the treatment. There were also some exclusions, which means that people couldn't participate if they had recently had rituximab or had a thymoma or had a thymectomy in 12 months, for example. So the primary objectives of this study, as I mentioned, were to study the safety and tolerability of Nipo, and then to evaluate its efficacy as measured by the Activities of Daily Living Scale, the MG-ADL, which many of you are familiar with, but I'm going to show this on the next slide. For those of you who aren't familiar with it. And so what we were looking for is what was the change from the time that people entered the study before they got the treatment until 8 weeks after being on treatment. And then there were a lot of other secondary and exploratory objectives, which are to look at the strength of patients as assessed by this QMG score, which is something that's rated by the clinicians at the bedside and with some other special equipment. We also wanted to look at look at responder analysis. And so a responder would be someone that had a change in their clinical status that would be considered clinically meaningful. And I'm going to show you an example of this on the next slide. So a change in their Activities of Daily Living scale that most patients and clinicians would say, that means something, that's relevant to patients. You know, if we can just change like the number and the total score, but for a patient, it doesn't have an impact on their daily life, then that's probably less important. And we also wanted to look at a durable response. So how long did the response last after it was achieved? If a response only lasts for a week or two, well, maybe that's helpful, but we would like to see that it's durable, that it persists for a long period of time when we treat patients. And so here that's defined as you had to have a 2-point change for four consecutive weeks during the 8-week treatment period. And so here's the ADL score, which has 8 different items associated with it that are scored 0 to 3. And a higher score means more symptoms or more problems that the patients are having. And the highest score that you can get is a 24. This scale is used very commonly in almost all myasthenia trials because the FDA that is ultimately going to approve a drug prefers this as the outcome measure. So let's go through a hypothetical patient here. So this patient that we're going to go through has a baseline ADL score of 8. And in this case, they have fatigue when they are chewing. They get weakness with even solid foods. They have shortness of breath when they're at rest, so just sitting. And they have double vision that's every day, but it's not constant. And they have eyelid drooping every day, but it's not constant. So now what we'll do is look at what happens if we improved the ADL by 2 points. So we go from an 8 to a 6. What might that look like from the perspective of the patient? So in this example, we have a patient that goes from shortness of breath at rest to with exertion. And so this might mean that a patient, had very limited ability to do activities around the house, household chores, do things with their kids or grandkids. And now, potentially if it's with exertion, you may be able to do more in your daily life. And then here, the other change in this patient was they went from double vision that was daily to it's happening still, but doesn't happen every day. And so you can think about how that might be considered an improvement for patients. So what I'm going to show now is the data through week 8 for the primary outcome of the study, which is this change in ADL score. And we'll also look at the safety data that's available. And the other thing that's important to note about this is that this study was kind of wrapping up right when COVID-19 was starting to become very active in the United States and elsewhere. But for the primary outcome here, the ADL score, the trial was not affected, and they were still also able to receive the safety data that they needed. So here are the key findings. Nipo was efficacious at the doses that were tested, and there were significant reductions in the IgG levels in the blood that was measured, and also in the acetylcholine receptor antibody levels as well. And the amount of reduction was correlated with the clinical benefit that patients experienced. So the more IgG that was reduced, the better their experience with the drug, the more improvement that they experienced. And we'll show that coming up. In terms of safety, so far the safety looks good. Again, this is a small study and we need to study it more, but to this point, it's been very well tolerated. There were no infusion-related reactions. There were no adverse events that led to stopping of the treatment. And there were no severe or serious events that were considered related to the drug. And so an SAE, or a serious adverse event, would be something that either causes death or hospitalization or prolongs a hospitalization, et cetera. And that was not observed. And then in terms of dosing, they saw that there were improvements with monthly dosing, and that you could see that within 2 weeks of starting treatment. And that's, I think, important for patients as well as for clinicians. And overall, it suggests that there's a rationale for continuing to develop Nipo for treating patients with myasthenia. And in addition, they're also interested in looking at a subcutaneous injectable formulation. The current formulation is a rapid IV infusion. So development of subcutaneous formulation will give more options potentially for patients. So here are the 5 different groups. So there were 68 patients randomized, approximately 14 in each group. There was one person that dropped out in placebo because they got worse after they started the trial, but didn't immediately after getting 1 dose of the drug. Here are some. There were a few patients that did drop out or stopped treatment related to the pandemic, but by and large, most of the patients completed the whole study. So 93%. Here, there were some dropouts here in these groups. And if we compare the characteristics of the patient population at the time that they started the study, this is important because if there are major differences, it can sometimes limit how we interpret, how we can interpret the data. But you can see for the most part is that in each of these groups, it was fairly balanced. And so what's shown outside the parentheses is typically the mean score, the average score, and then in parentheses is a measure of variability, the standard deviation, or in some cases the percentage of patients. So for instance here, if you look at the percentage of female patients, it was essentially the same in both arms, and the age of the patients was very similar. There were some differences in terms of the duration of disease between the groups. But here, all of them, the average was well over 5 years in both. So, but this is something that, we'll continue to look at in future studies as well. In terms of severity of disease at baseline, it was fairly consistent across the groups, and most of these patients were mild to moderate in terms of their severity. And overwhelmingly, and not surprisingly, the patients had acetylcholine receptor antibodies. In terms of safety, so adverse events that happened after treatment, so overall, it was fairly balanced. So 81% had an adverse event in the Nipo arm, and 79% in placebo. No grade 3, so a grade 3 would be a severe adverse event. The most frequent ones were 11% headache, upper respiratory infection, and diarrhea. Here you can see the percentages in the placebo group as well. And I think it's probably more helpful to look at the percentages here, because you can tell that in the placebo group, there's 14 patients versus 54 we're looking at in Nipo. So the percentage probably helps us understand it better. And in terms of serious adverse events, there was one patient in the Nipo group that had a severe, that had shoulder pain, but on a previously operated shoulder, and that was felt to be unrelated to the study drug itself. And then there were two SAEs in the placebo patients, one that had a stroke and then one that had a serious worsening of their myasthenia. And so now let's look at the results on the ADL score. So this is looking at the durable ADL response. And if you recall, this is at least 2 points for 4 consecutive weeks. Okay, and so here that was achieved if you looked at all of the Nipo groups that were included in the trial, they were able to achieve that in 59% of the patients versus 15% in placebo. And that was a highly statistically significant result, at least for a trial this size. And then if we break that down in terms of the different treatment arms and the amount of drug that patients were receiving, you can see that there's a dose response. So the higher their dose, relatively speaking, the better that their response was to the drug on the ADL score. And these higher doses independently or separated out were also statistically significant. And this next slide is looking at how fast patients responded. And so you can, imagine if you're a patient, you obviously want to get better as fast as you can. And for the doctors, we also want the same thing. And in certain circumstances, that can become really important in terms of how we select medications. If we have someone that's not doing well at all, we don't want to choose a medication, or we're not going to consider a medication that's going to take 6 to 9 months to be able to show an effect. And so here, this analysis shows that this treatment seems to work pretty fast. Within the first couple weeks, about 40% of the patients had a clinically meaningful improvement or response over that period. And now this final slide is looking at the durability of the response here. And so this is a little bit complicated, but here we're looking at the change from baseline. And so you can, if you recall, a higher score is worse symptoms. So we're trying to reduce the score to lower it. So the more negative you are, the better that you're doing. And each of these colored bars are the different doses. And so you can see here 1 month approximately after starting treatment, there was, a 1.4 reduction in the ADL from baseline in the placebo group, and then 1.7 in the 5 milligram group, and then 2.9, 3.9, 3.1. And so you can see that the 60 milligram doses had the biggest effect. So within a month, the 60 milligram doses resulted in the most improvement for patients. Okay, but what about at day 57? How long did the response last, or how durable was it? So again, this is presented the same way. And I think the key thing that stands out right away is that if with the 60 milligram single dose, although it had a very big effect early on, it didn't last, or it seemed to be wearing off by the time you got to day 57. Whereas if patients who continued to get doses, so this would be every 4 weeks, every 2 weeks, those patients did much better. So, and that the response was sustained when they were on those treatment regimens. And so these results suggest that we can't, if we just give patients one dose of the drug, we can have a response pretty early, but it's not going to be persistent unless we continue to dose them with the treatment. And so now there's a lot of work that's ongoing. If we're going to continue to the next step, to the next stage, to a phase 3 clinical trial, is to determine what that dose would be for the phase 3 study that provides the best efficacy, but also maintains the safety, and hopefully is still convenient for patients and minimally disruptive for their lives. So that's the end of the presentation for this particular clinical trial. I mentioned there are a lot of other endpoints and outcomes that are of interest and that are part of the clinical trial protocol. And a lot of that data is still being analyzed. And so that will obviously be really important to further enrich our understanding of how this drug is operating in the MG patient population. So now I'm going to transition and show, just introduce people to a project called CARE-MG. And so CARE-MG stands for COVID-19 Associated Risks and Effects in Myasthenia Gravis. So this is a study that is being conducted by an international group of investigators. And the goal here is to understand the effect of COVID-19 on patients that have myasthenia gravis. To this point, this is obviously a very rapidly changing environment, and our understanding of COVID-19 is changing month to month. But we currently have very little data that we can give to patients to educate them and to help us as clinicians decide the best way to manage patients in the current pandemic. And so what we are doing is creating a registry to collect information on patients' experiences that have had COVID-19. And this registry is completed by clinicians, so the treating physicians that treat the patients. And this data gets entered into an online database. It's all, there are no identifiers that are included, no names and dates of birth or anything like that could identify individual patients. And so we're collecting that information so that we can understand what the impact of COVID-19 is on patients who develop the infection. And so there's a steering committee that is worldwide, so North America, Europe, and Asia that have put this together. And we are continuing to collect cases from all over the world, and we're trying to get the word out to as many people as we can so that we have as much data as possible, so that we can, understand this situation as completely as we can, understand, what changes we should make or shouldn't make in terms of managing a patient who happens to get COVID-19. So with that, I am going to stop there and see if there are any questions from the audience.
Hi, thanks, Dr. Guptil. We really appreciate you reporting out on the study very much. We have a lot of questions, so
Okay.
I'm going to facilitate and field some questions to you. And I don't think we'll be able to get to every single question, but what I can tell folks is that we will make sure that so we have everything that's in the chat, and we will make sure that every single question gets answered, and we will post it on our website. We'll send some type of communication out to notify you that that's been done, so you know when that's done. It'll take a little bit of time, but we'll work with Dr. Guptil and certainly the Momenta team to make sure that every question is answered. So with that, Dr. Guptil, I'll start asking you some questions now.
Okay.
When people enrolled in this trial, did they have to stop taking their current therapy?
Yeah, that's a really good question. This is a very common question that we get from patients who are considering a clinical trial. And the answer is no. So the patients who were already on treatment, as long as they weren't on one that wasn't allowed, and that can be very different depending on what the trial is. But for most of the common treatments that patients take, like mestinon or pyridostigmine, prednisone, mycophenolate, or azathioprine, is that those are all allowed in the clinical trial. And so typically it's whatever the patient is on at the start of the trial, those doses and treatments need to stay the same throughout the entire period of being in the trial, at least the initial phase. And that's a safety thing, right? For a drug that we don't know whether it works or not, it would be dangerous to take people off of medications, particularly if there's a placebo arm where they might not be getting an active drug. So the expectation is that if you're in a placebo arm in one of these trials, you're continuing to take your regular medications, and you would expect to be stable. It would be your normal situation, but you wouldn't get better or hopefully any worse during the treatment period. So we're looking for a benefit above what your normal treatments are offering you or providing you.
Thank you. Can you just talk a little bit about how people found out about this trial?
Yeah, so that's another, that's a good question. So most of the time in these, for trials like this, and particularly in the myasthenia gravis space, is that you would normally learn about these trials from your treating physician, so your neurologist. Is that if your neurologist was taking part in the study, they would obviously know about it, and if they thought you might be eligible and interested, that they would then approach you to participate in the study. So this approach works, of course, because you know the neurologist, and you have a relationship, and they know you well, and whether or not you'd potentially be eligible to participate. The issue is that it doesn't get the word around to everyone out there that might be interested to participate. And so, there's a lot of effort now to try to improve the advertising or the knowledge about what's going on. And this is an example of that, is doing educational programs like this. So now you guys know about this particular product, Nipo, and that if there's a phase 3 study, you might be looking out for it in the future, and you'll get similar information from the future clinical trial updates that will be provided as part of this series. In addition, the MGFA is also doing a great job in providing information about all the opportunities that are available to patients on their website and I believe through their social media accounts as well, so that people know what's going on in the field. So the MGFA can be a very helpful resource to know about at least what trials are out there, to try to start that discussion. There's also a website that you can go to that it can be difficult to kind of sort through it, but it's called clinicaltrials.gov. And it's a website that posts all of the phase 2 and phase 3 studies that are active. And you can just search for myasthenia and you can see everything that's ongoing out there. So that can be another resource for people. And then finally, if you're an investigator within the study, like I am, I try to tell as many people as I can about the trial so that I can hopefully, get as many people interested in it who might want to participate. So a lot of us will send letters to other neurologists in the area, letting them know, hey, we've got this clinical trial. These are the types of patients that we're looking for. If you have someone that might be interested in this, let me know, and we can see if they'd be interested and eligible to participate.
Okay, thank you for certainly sharing that website, the clinical trials website, because that's really great for our community members to have. So our understanding is that this was an interim analysis. So can you talk a little bit about why you all did this, and will it be different from the final analysis? And then what other information do you anticipate will emerge from the final analysis?
Mm-hmm. Yeah, these are great questions. So the interim analysis in this particular case was really to understand or get a sense of whether the safety and whether or not the drug appears to be working on that ADL score. And so that information is really helpful to the company because it really becomes a quote, go, no go decision. You hear this a lot in the clinical trial world, is that, if it looks like there's a safety issue, then you're probably not going to move forward with it. And if it doesn't appear to be impacting patients at all, helping them, then you're also probably not going to move forward with doing another trial. So having at least a preliminary impression of the safety as well as the efficacy of the drug can help the company make decisions as to what they want to do next. And so, as I showed very early on, this process takes years and years to do. So in a lot of cases, the company may do this type of analysis relatively quickly at the end of a trial to get a sense of whether or not it appears to be working. And then if so, then they can start their plans for the next phase to do the phase 3 study. And the planning for the phase 3 study takes months and months, up to a year sometimes, to get straight. So, the company's very motivated to move things forward as quick as they can. And obviously, if patients can benefit from it, I think most of the patients would be for that. And the clinicians want to move the therapies forward as fast as we can. So that tends to be the main rationale for doing it. In terms of it, so with that being said, there's tons of data that are collected during these trials. And so it takes obviously time to go through that data and to understand it and analyze it fully. And so that process can take many months to do that. So those are the types of things that are still ongoing as part of this trial, is to get a deeper understanding of the effects of the drug in this particular trial. So in terms of what to expect down the road from this particular study, we wouldn't expect there to be any changes in what's been reported already in terms of the ADL scores, because that's been analyzed. The safety data, that's been analyzed, at least through this point, day 8. What will be added is to round out the picture. I mentioned the QMG score, so that'll be something that'll be added to the picture, as well as other endpoints and things that were looked at as part of the clinical trial conduct.
Okay, thank you. Is there anything else that you could add about the delivery of the drug and the dosage specifically?
Yeah, so the trial, so I presented that during the course of the presentation. So there were 5 different arms. So and they were different doses of the drug to try to get a sense of how much drug patients would need to improve. And it seems to indicate that one is that all the doses that were tested seem to have some effect, but the more drug that you got, basically it appears the better that you're going to do in terms of your myasthenia. That's the type of additional data analysis that's ongoing and work to try to figure out, based on all this data from these different arms, what is the best dose to move forward to test as the pivotal trial for this particular drug to bring it into phase 3. There was a second part to that question, wasn't there?
I asked about the delivery of the
drug. Oh, the
delivery.
Yeah. So that's so it's an IV treatment. It's a rapid infusion. And so I think it's 15 minutes to administer the drug, 15 to 30 minutes, I think, depending on the dose. So it's very rapidly infused. And as was presented in the slides too, the company is also working on a subcutaneous formulation. So you don't need to give it through an IV. You can give it like an insulin shot, sort of. But that's not out there yet.
Okay, thank you. All right, one more question about this study, and then I do want to be able to ask one question on COVID, on the other piece of the information that you shared. So you see a lot of MG patients, and what is one of the things that you found most interesting about this trial and its results?
So I'm going to broaden this a little bit to not answer that specifically from the standpoint of this trial, but to look at it from the perspective of the class of treatments that are being developed against the FcRn receptor. And so I think what we're seeing, some of you may be aware of this, is that there was another trial results came out yesterday for another trial from the company Immunovant that's also developing one of these FcRn therapies that was also positive. And so what we have now is a fairly large body of evidence that suggests that this is a treatment strategy that works. And so, we have Argenex, which is one company that has developing efgartigimod for generalized myasthenia that has finished their phase 3 study that was also positive. We have UCB Rod that's working in this space, and Momenta and Immunovant, and I'm probably leaving people out there. But so I think the biggest thing for me is that we have quite a large body of evidence to suggest that this is going to be a treatment that's going to work for patients. And so far, what we've seen is that it's also quite safe. And the final thing to say about it is that what's really, I think, nice about these treatments are that if you think about the effect of steroids and some of the other oral medications that we use that create a more widespread suppression of the immune system function, is that these drugs are much more targeted, and a lot of the other aspects of the immune system are untouched. And so that will, I think, could deliver some real benefits to patients in that they're these very targeted treatments to approach their disease compared to what we've had in the past.
Okay, thank you very much for that. I know we're at the top of the hour, but there is a question on COVID, and I know this is top of mind for a lot of people. So I just want to ask this real quickly. Michael asked a question: how much more dangerous is it for MG folks to be out and about with COVID-19 in the area? Should we be wearing face shields, or is a mask okay?
Okay. Yeah. So, this is the type of question that we're trying to answer with this registry, and to be able to say with a lot of certainty what people should be doing. So right now what we can say is that patients should be following the precautions that the CDC have proposed for preventing transmission of COVID or getting it. So all of those things, the face mask, social distancing, frequent hand washing. So all of that is really important. I don't know about face shields and the impact that might have. I think that the bottom line is that you need to be really careful. So to date in the registry, there have been 70 cases that have been reported, and the outcomes for patients tend to be pretty severe, is what we're seeing. So it seems to be that it affects MG patients more severely than the average population. So, I'm strongly advocating for my patients that you have to take all the precautions and, as much as you can to limit your exposure to people until we can get that vaccine to become available.
Okay, thank you. I appreciate you answering that one last question.
Yeah, and one other thing I'm sorry to mention is so flu vaccines. Everyone should be getting their flu vaccine injection in the fall, because that's going to be a major confounder in terms of patients in the winter time getting respiratory illnesses. Is it COVID or is it the flu? Now, we can't predict whether or not the flu shot is how effective it's going to be, but assuming it is, it could be really helpful to not have to contend with that issue as well.
Just trying to come off mute.
Yeah.
Dr. Guptil, thank you so much. Thank you so much for delivering this information today and for joining us, and certainly for the support that you give us all the time. So we really, really appreciate it. I also just want to thank everyone else for listening in today. I know that it helped all of us, and there was tremendous value in this. We certainly would not have been able to kick off the series today if it were not for our great partners over at Momenta Pharmaceuticals. So I do want to congratulate them on being able to share this good news and the due diligence and hard work that they certainly have been displaying as a team. So with that, I want to thank you all once again, and you'll definitely be hearing from us with the next session in the series. And as soon as we have some new exciting information to share with you, we will be bringing that your way. So thank you so much, and have a good rest of the week.
Great. Thank you, everyone.